Well, good afternoon and welcome to the fMRI course lecture on non-invasive neuromodulation applications. My name is Dr. Holly Lisanby, and I'm joining you virtually for this webinar. As we go along, please feel free to use the chat tool built into WebEx to send me any questions. And I would ask you to mute your phones so that we can control the avoid any audio interference. I'm sharing my slides, so hopefully you can see those. So again, my name is Holly Lisanby. I'm the Director of the Division of Translational Research at the National Institute of Mental Health. I am also the Director of the Intramural Research Program Unit on Non-Invasive Neuromodulation at NIMH, and I'm a co-lead of one of the Brain Initiative teams at NIH. You've already heard from some of the members of my unit earlier in this course, in particular Drs. Tom Radman, Zi Deng, and Bruce Luber. And as you've seen reflected in their presentations, we are a multidisciplinary team bringing together expertise from the area of engineering, neuroscience, and clinical psychiatry. So I'm the psychiatrist part of the team, and today's presentation will be about applications of non-invasive neuromodulation tools for therapeutic purposes and also as tools for discovery to understand the brain basis of disorders. So without further ado, I'm going to start by talking about what the promise is of these non-invasive neuromodulation technologies for therapeutic applications. And specifically, when we think about what brain stimulation can offer, take, for example, in the field of psychiatry, we have two major modalities for intervention. We have psychosocial interventions and we have psychopharmacology. What non-invasive neuromodulation brings is a third pillar to these aspects of modern clinical practice. Now, in addition to drugs and talk therapy, we have device-based therapies that can be used in combination with these other two approaches, and it really expands the therapeutic potential for our patients. And in a very specific way that's exciting for researchers, and that is it expands our ability to translate knowledge of neurocircuitry underlying brain-based disorders into targeted treatments. And we are at a stage today that this is already a reality. Take, for example, our knowledge of the neurocircuitry of depression, which has been used to develop an FDA-approved treatment for depression using transcranial magnetic stimulation, or TMS. And so this is actually one of the first translational demonstrations of the translational impact of studying neurocircuitry in the way that it can bring forward a new therapy. Another example would be the greater knowledge that we have of neurocircuitry underlying, in this case, addictions, could inform what brain areas could be therapeutically targeted to not only change activity at the site of stimulation, but also to change activity at connected regions of the brain to modulate the circuit in order to have improvement in different aspects of the syndrome shown here. So what's the current state of the art for clinical non-invasive neuromodulation? Well, today we have clinically effective FDA-approved treatments. Specifically, and this is not new, this has actually been since the early 30s, we've had electroconvulsive therapy, or ECT, which is effective. We continue to do it today because it is unparalleled in its efficacy for severe depression. And shown here are the likelihood of responding to antidepressant medication. So in the green bars you see, after you have one medication exposure, the likelihood or the percent of people with depression who respond to that first medication is actually less than 50%. When those patients fail their first medication, they move on to a second medication, and their likelihood of responding to that second medication is about 10%. When they fail that one, their likelihood of responding is even less than 10%. So you can see that with medications alone, once patients have demonstrated to have treatment-resistant depression, there are diminishing returns from making one med change to another. Contrast that with the powerful antidepressant efficacy of electroconvulsive therapy, which typically has response and remission rates of upwards of 80%. And that's even when people have failed to respond to medications. And so these are the most refractory patients, and they are responding the best to this device-based therapy, ECT. So we continue to do ECT today because it has an unparalleled clinical efficacy. We also continue to do it because it has an exceptionally rapid speed of response, especially in life-threatening conditions such as suicide. Here we're looking at the cumulative percentage of patients in the blue line and then the percent at each treatment that are continuing to experience active suicidal ideation. So after the first treatment, 30% of those patients have a resolution of their suicidal ideation. By the second treatment, it's half of the patients. And by the third treatment, it's about 2/3 of the patients who have had a complete resolution of their suicidal ideation. So you can see that in the matter of a week, the majority of patients are no longer thinking of killing themselves. And this really reflects not only how life-saving ECT can be, but also how rapidly it acts, which when we're dealing with life-threatening depression, the rapidity of action is really essential. But today, we have less invasive alternatives that I'm going to be talking about for much of this webinar, such as transcranial magnetic stimulation. And you've already heard from my colleagues about the physics and the engineering behind TMS and how it can be used in neuroscience investigations. And I'll be focusing today on the therapeutic applications of this. So it's less invasive. It uses magnetic fields to non-invasively induce electrical currents in targeted regions of the brain. And this is a picture of the actual clinical setting where it's given. So the person's not under anesthesia. It's a medical office-based setting. A seizure is not being induced. And the depression treatment protocol, they come in daily for about a 40-minute session. And over a period of 4 to 6 weeks, there's significant reduction in depression scores. And this is available in insurance in many areas is covering it. I was showing you the first device on the market. The second device on the market used a different shape coil, and you heard from my colleague Zi Deng about the impact the coil design has on electric field distribution. This is the DTMS coil or helmet coil, which is also on the market and approved for depression as well. And there are other newer devices that are at various stages of development. So the current state-of-the-art today of clinical application of non-invasive brain stimulation is that we have these safe and non-invasive tools such as TMS that can be used for mapping brain behavior relationships in an image-guided fashion. As my colleague Bruce Luber already presented, you can use fMRI results to guide the TMS delivery using neuronavigation to stereotaxically apply TMS to nodes within networks to ask what happens when we change activity within the circuit, what happens to behavior, what happens to physiological response. And we can precisely time the delivery of the TMS during the conduct of neurocognitive tasks, such as in this example where the subject is using a button box to respond to cues on a computer screen, and these little antlers are for the neuronavigation, antler on her headband and also on the coil. So we can precisely in space target and in time couple the TMS with cognitive behavioral task performance. And we can also do simultaneous physiological readouts, such as using fMRI or event-related potentials to understand how the TMS is exerting the changes that we observe in behavior. So here's an example of putting the TMS coil inside the head cage in the MRI scanner, and the TMS device is in the fMRI control room, and we very precisely time the TMS pulses with the echoplanar imaging acquisition so that we can look at the bold response to the TMS paradigm that's being used to exert cognitive change. And as my colleague Bruce already presented, when we are clever about our targeting, we can get transsynaptic effects and see changes deep in the brain, regions that are connected to the superficial regions that we're targeting. You can do that with simultaneous imaging. You can also use event-related potential shown here, which was among the first demonstrations in humans that TMS, when given in a repetitive fashion, can actually potentiate a brain response and result in an effect that is similar to long-term potentiation. And so that you can focally modulate plasticity in the brain. Now, the fact that you can do that in cognitive neuroscience studies means that you could also do this, if we're smart enough, in a clinical context to figure out and identify where are the plasticity deficits and then target the TMS coil to remediate those in a clinical setting. We are also at the state where we have affordable and portable technologies that are on the horizon that also have excellent safety profiles, such as transcranial direct current stimulation or transcranial alternating current stimulation. Now, these are not FDA approved for treatment, but we have done a number of large-scale trials with TDCS and some additional new studies with TACS, suggesting that these may be, if we're smart enough about how to shape the field in space and time the delivery to modulate functioning of circuits, may have therapeutic potential as well. So this is what the standard TDCS looks like, and this is the device that's modified specifically for clinical trials that has blinding for operator as well as for subject in a sham mode. And then there are multi-electrode arrays or high-definition TDCS that allow us to have specific multiple anodes and cathodes to shape the field more precisely than you can do with the two large electrode array, which the original TDCS device was designed as. So with these high-definition TDCS, you can model in silico what amount of current you want to inject and this is a 4-to-1 array, so the aqua colors are showing you that we're injecting a current of about minus 0.5 milliamps, and the red electrode is the current return electrode. And with these arrays, you can then model what your electric field distribution would be in the brain and use this technology to really tailor the field to the areas that you want to target, likely informed by the structural and functional imaging of each individual subject. So in addition to all of this, we have a growing industry base, and this is supported by FDA approval, such as for TMS, and supported by insurance reimbursement in many regions of the United States, including right here in Maryland. So there's a lot of development in a short period of time with clinical application of neuromodulation. And there's, in addition to all that, a strong consumer base. People are interested in this. There's a do-it-yourself community and growing demand for non-invasive tools that can be used clinically. Regarding the do-it-yourself community and the lifestyle companies that are popping up and entering the picture in this realm, all you have to do is Google Neuromod and DIY, and you'll find ways to build this. So what's wrong with this picture? It sounds like I've been leading up to a very exciting state of the art of clinical neuromodulation, but where are the research challenges? Where are the gaps? What are we not getting in terms of maximizing the clinical utility of these technologies? So I've talked about two technologies, ECT and different forms of TMS, and there are limitations to these. With ECT, it's highly effective, and it's arguably underutilized when we think of the number of people that could benefit from it. Why? Because of side effects, because of cognitive side effects of ECT. TMS is a safer alternative, and although it does not have the cognitive side effects that ECT has, its efficacy falls short of what we see with ECT. Here's the pivotal trial that led to the FDA approval of TMS, and in the blue bar, you see the response rate with active TMS in blue and sham in the gray. And you can see that after 4 weeks of treatment, there was a separation, and after 6 weeks, a significant separation. But the overall response rate after 6 weeks of daily treatment was still less than 25%. And I've already told you that the overall response rate to ECT was upwards of 80%, so not nearly as close. So ECT remains more effective. And the efficacy of TMS is really closer to what we typically see with antidepressant medications, which I've told you, once a patient has shown that they're non-responsive to antidepressant medications, the benefit of making a change to a new drug class has diminishing returns. Now, it's not just about the non-invasive approaches, but the invasive approaches like deep brain stimulation we thought had great promise because we could put the electrode in deeper regions of the circuitry underlying depression that are not accessible to the direct effects of TMS. As you learned from my colleague Zi Deng, the electric field strength with TMS drops off exponentially as a function of distance. But with deep brain stimulation, you can put that electrode exactly where you want it to go. Yet 2 major industry-sponsored DBS trials were halted because of a futility analysis showing that further subject recruitment would not likely show a difference between the active and the sham. And this was very disappointing. So there's a lot of promise in the field of neuromodulation, but there are also challenges. So to highlight them, improving the safety of ECT, improving the efficacy of TMS, and figuring out how to better target deep brain stimulation in space and time all represent research gaps that are actively areas under study. So why is it that we have these limitations? What has led to this promising and yet a situation where the promise has not been fulfilled? That's what this talk is about. So what are the limitations of the current non-invasive neuromodulation that impacts efficacy? Well, one is variability in practice. This is certainly true in the case of ECT. The way the treatment is done varies depending on where you go. So if you get ECT at Duke, or if you get ECT at suburban hospital. The standard practice differs as a function of region and how the doctor was trained. And we already know in the field of ECT, the way you do the treatment determines the outcome. So you want to have better control and understanding of these practice parameters that influence outcome. Well, and we really like to be able to optimize the dose to improve its efficacy, but it would help if we knew how it worked, right? In order to optimize the dose, to improve efficacy, you need to know what is the mechanism of action and focus on that. That is a missing link in our research knowledge to understand mechanism that could then be used to optimize dose. So our limited knowledge of mechanism of action at a cellular and circuit level has limited our ability to inform and optimizing dosing strategies. So it's really a black box that we're treating it like. Somewhere within this black box are the clues to the mechanisms of action, the duration of action, optimal dosing, and measurements of target engagement. Is the treatment really engaging the therapeutic target? And the goal of research and where we are now with our funding priorities is to open up that black box and discover how the most effective treatments that we have work so that we can make them safer, to discover how to improve technologies such as TMS to make them more effective, and to discover how to optimize the targeting of deep brain stimulation approaches to improve efficacy. So these are I'm highlighting some key research opportunities, and for those of you who are attending who are thinking about how can you have an impact, what could you do with your research career to help us make progress in improving the clinical application of neuromodulation to improve brain health and mental health in particular, these are some of the opportunities. So, for example, it would be helpful if we could determine the fundamental dose-response relationships. How do we tweak the dosing of the stimulation to have desired effects on physiology and circuit function? And to employ expertise and equipment that is uniquely available within, for example, the NIH and NIMH intramural research program, such as the availability of non-human primate models, where one can directly study the impact of stimulation on measurements of circuit function. Also, we have clinical populations that can be intensively studied in the clinical center. The clinical center, as we know from the recent Discovery series, is really a crown jewel in the nation's investment in first-in-human research, and to take advantage of those resources to advance these technologies by doing the first-in-human studies to improve their efficacy. We also have available at NIH important physiological tools to understand the impact of the stimulation on brain function, such as magnetoencephalography and receptor PET, and technologies to put the stimulation inside of the imaging facility. This is a technology called perturbation imaging, where you stimulate while imaging, such as with TMS, fMRI, interleaving. So these are all fantastic research opportunities that are available to you as students and researchers at NIH. There are also opportunities to do collaborative studies across disciplines. As I've already introduced in our non-invasive neuromodulation unit at NIMH, we are a multidisciplinary program bringing together expertise in engineering and neuroscience and clinical trials and psychiatry in order to advance and leverage these technologies. So if you are contemplating doing an experiment using one of these technologies to address a brain health or mental health related question, we can be effective partners to help you answer some key questions about how to most productively use the tool to ask the questions that you have. And to leverage the depth of expertise that's available here at NIH in the physiological effects of non-invasive neuromodulation in neurological and psychiatric applications to address relatively less explored complex neurobehavioral conditions in psychiatry. So it's really an ideal time to be working in the field of clinical neuroscience and intervention development in psychiatry. And because we have these innovative tools, because we are working across disciplines, and all of this can be informed by the types of electric field modeling that my colleagues Zidane and Tom Radman presented earlier. So some advice that I have as you are thinking about how to incorporate these tools into your research programs are to not treat these tools as off-the-shelf black boxes. Remember, I talked about the mechanisms of action and the targets being within that black box, and so many groups may be limited in their ability to have impact because of treating the tool like a black box, as if we knew everything we needed to know about it, when in fact the mechanisms of action of the tool itself are an area of active study. There are also opportunities to think about innovative novel paradigms, different ways to stimulate, different ways to approach your analysis, and particularly using engineering tools of signal processing to understand the physiological response to the stimulation, and novel paradigms for perturbation imaging, so combining the simultaneous stimulation with the imaging to advance our understanding at a mechanistic level. And I'm going to go into some depth on this next point of advice, which is to pay attention to precision in the dosing. And I'm going to introduce three aspects of dosing: the dosing in space, dosing in time, and dosing in context. So this refers to the spatial distribution of the electric field in the brain, the temporal dynamics of your stimulation paradigm, and the context in which the stimulation is delivered, which means what is the brain doing at the time you're stimulating. Attention to all three is essential to optimize the use of these tools to address the research questions that you have, and it's also essential to make inferences to understand what you've learned from your research project when you're analyzing your results. So let's go a little more in depth now about these three dimensions of dosing that are relevant for the next generation of neuromodulation applications. So starting with, of course, we're with eclipse fever. I thought it would be nice to pull a photograph from the Hubble telescope, but this is meant to reflect the spatial distribution of the electric field, which is determined by the shape of the coil, its orientation relative to the target cortical structures, and the individual anatomy. So the structural MRI, the tractography, the diffusion tensor imaging, all of this goes into identifying the spatial distribution of the electric field in the brain. The temporal aspect of dosing, and I'm a big Dolly fan, so the temporal aspect refers to the rate of change of the electrical pulses through the stimulating apparatus, whether you're dealing with electrodes or, in this case, a TMS coil. So that rate of change of the electrical current through that coil, which determines the temporal aspects of the dosing. So the pulse width, the pulse shape, the pulse directionality, the frequency, the duration of the train, the interval between the trains, all of this will influence the outcome. And then context. In this case, of the far side, where you're telling the cat never ever think outside the box, context matters in terms of litter boxes, and it also matters in terms of TMS study design. And so the context in which TMS is delivered is a social one. There's more than one person in the room. So you've got the experimenter who is manipulating the coil and interacting with the subject. You might have a task that's underway during the stimulation. The subject may be undergoing a pharmacological manipulation, such as taking a medication or having a pharmacological challenge during the stimulation. All of this goes into the context. We know from physiological research that the brain's response to stimulation depends on what the brain is doing at the time of stimulation. If you think of moving your finger, when you're getting your primary motor cortex stimulated with TMS, the motor response of the finger twitch will be increased. Now, if you think of other things, for example, if you're trying to remember 3 letters that were just shown on the computer screen when you're being stimulated, this may enhance your cognitive function or disrupt it depending on how and when the TMS is applied. And as people are joining, let me just mention that I have the chat tool open, so please feel free to interrupt me with the chat tool with any questions that you might have. Otherwise, I'm just going to continue with my presentation and sharing the slides. So I think of this as the where, when, and how of non-invasive neuromodulation. And when we think about optimizing the clinical applications of these tools, it's important to understand all 3 dimensions. And I can say very rarely are all 3 dimensions of dosing attended to in study design, and I mean even in the pivotal trials that led up to the FDA approval, and in experiments that you see published on or advertised in clinicaltrials.gov. And so there's a great opportunity for young researchers entering this space with a sophisticated understanding of the neuroscience impact of these aspects of dosing to really advance and propel our field. The next bit of advice that I have for you when you're thinking about how to apply these tools is to think about targeting circuits, not sites. So we know that when you stimulate one site of the brain with TMS, you are actually stimulating a circuit. You're stimulating 1 and 2 synapses down the chain. You're stimulating and changing activity at transsynaptic locations. We know this using TMS fMRI and TMS evoked potential. And yet when studies are designed, typically people think, I want to put the coil where the fMRI blob is, without thinking through, well, actually, it's not a discrete location, but it's trying to optimize the targeting of the TMS to achieve the maximal effect on the distributed network, which is your target of interest. And that's true whether you're trying to study a neuroscience application of TMS or whether you're trying to develop a treatment. So thinking about the transsynaptic action of TMS, that's nicely illustrated when you put the TMS coil inside the fMRI scanner and you image the bold response. And here we're looking at distal activations in blue and deactivations in red induced by single pulses of TMS given to the prefrontal cortex. And you can see a lot is lighting up here that's quite distant from the prefrontal cortex. And so obviously it's stimulating a network, and knowledge of this network is important to optimize how you're delivering this to affect the circuit that you want to study, and it's also important to interpret the results of your findings. So putting the coil on the prefrontal cortex and finding a change in an aspect of behavior does not mean that the prefrontal cortex was responsible for that behavior change, when in fact the TMS is affecting regions of the brain outside of the prefrontal cortex too. The next point of advice is to employ E-field modeling, so modeling of the electric field distribution in the brain induced by the TMS, coupled with connectivity mapping to know where you actually stimulated. And by connectivity mapping, I mean not just three-dimensional models that take into account the white matter tractography, but also understanding the strength of connectivity from one site to another. And some very nice work from Alexander Opitz has demonstrated why this is important. At the top row, we're looking at coil locations, which were applied based on this 2x2 grid, trying to target prefrontal cortex. And on the next row is showing what the E-field distribution is, so the electric field induced underneath the coil. But the bottom figure is showing you what that blob that was stimulated was connected to by applying the E-field model in B onto the human connectome data set on the lower part of the slide, so that you can see when we stimulate prefrontal cortex, where are we also likely to be affecting based on the individual connectivity. And you can see that it's quite broad and distributed network, but also that distributed network is influenced by factors that people don't always control in their experiments, namely coil orientation. Shown in this slide demonstrates how when you rotate the coil from 0 to 90 degrees, the E-field changes somewhat, but dramatically changed is the transsynaptic or distributed connectivity map of where your stimulation would be influenced in terms of the connected regions of the brain. And so taking that extra step, not just to do E-field modeling, but to apply that to connectivity mapping will be very important in deciding which coil orientation you want to use for your experiment, individualizing that based on the individual anatomy, and then understanding where you're also having transsynaptic action as well. This approach has been applied to the clinical trials that use TMS in the treatment of depression. And you see here across studies that are enumerated that did apply TMS to prefrontal cortex using different approaches. And the colorized balls show where the average coil location was along the middle frontal gyrus. And this connectivity map shows, so basically it's color-coded, showing, for example, the experiments in the blue, which is the Herbstman, for example, has a very different connectivity pattern, network connectivity pattern, than some of the other experiments which were trying to target the same region, but using a different orientation, slightly in different location, getting quite a different network. And so when you're doing a meta-analysis to try to understand from the published literature, what have we learned about the effects of TMS to prefrontal cortex, acknowledge that it's like a shotgun approach here. There's quite a degree of scatter. And this is a challenge for making productive use of meta-analyses, which is why we really advocate when you're doing studies with TMS that you report all of the aspects that go into the dosage. So not just saying that you stimulate DLPFC, but exactly where and how and what your coil orientation was and what the human connectome data says you were connected to, so that future meta-analyses can be more meaningful. So a next bit of advice is to use, so the second to last bullet point was really about targeting, to model your field and understand where what you're stimulating is connected to. And the last bullet point here is to measure the impact of the stimulation. And that's what's meant by target engagement. So not just knowing, here's what I injected into the brain, but knowing here's how the brain responded. So if you stimulated an area of the brain and you measure just a behavioral response, you may not know what caused that behavioral response. You can't necessarily assume it was where you put the coil. It might be something that was downstream. So using measures of target engagement where you've got the perturbation imaging or TMS evoked potential can help you demonstrate whether you actually stimulated your direct or transsynaptic target that you were trying to reach, and helps you interpret your results, both your successes as well as your failures. And so I'm going to give an example of target engagement from some of our work, and some of this was reviewed by Bruce Luber, but I'm taking a little bit different orientation to describing it. And as I do this, let me just remind you, I do have the chat tool open, so please feel free to interrupt me with any questions using the chat tool. And please send your question to all. I'm not actually the host of the webinar, so please send any questions copied all. So target engagement, here's an example where working memory was the domain of function that we were targeting. And we were using this task, which is the Sternberg delayed match to sample task. The subjects were given 1, 3, or 6 letters. Then the screen went black for a 7-second retention period. Then they were showed a probe, and they were asked with a button box whether the probe was in the study list or not. And we had mapped this with fMRI. And our target was a network of brain areas which were more active in subjects who were better at performing this task, specifically subjects who were resilient to the adverse effects of sleep deprivation. So this target was our neural reserve and compensation circuit, something that helps explain individual differences in terms of their susceptibility to the adverse effects of sleep deprivation. And so we gave TMS to a node within this network that we thought was important for neural reserve, as well as a nearby control site. And our intervention was not just the TMS, but TMS with simultaneous working memory training, because we understand the brain's response depends on what the brain is doing when it's stimulated. So our intervention was really a combination therapy, activating the circuit with the working memory task performance, and then neuronavigating the TMS to each individual site that's activated by that task in that subject, and giving it at the exact time that that node in the network is being utilized. So it's not only for spatial targeting, but also temporal targeting. So time locking the TMS to the phase of the task when the area of the brain that's stimulated is being used. So that's the fMRI guided simultaneous TMS and task performance. So the two components is the neuronavigation and the simultaneous delivery. And I'm emphasizing that point because this is really a gap area. Most projects don't do online task performance with the goal of enhancing function. Typically, they're using the goal of disrupting function as a mapping tool. Here, this is really an intervention development tool to enhance function. And so that simultaneity is really important. And then we used as a demonstration of target engagement the expression of this compensation network. Using fMRI afterwards, did the degree to which we improved the utilization of the compensation circuit explain individual differences in the effect of the intervention on outcome. So we predicted that there would be a shift in fMRI network expression during the task performance. And this is really illustrative of a translational trajectory strategy where we started with trying to enhance normal function and then moved on to trying to remediate function when it is impaired in healthy volunteers by an intervention that disrupts function. That's why we use sleep deprivation. We then used it to remediate the function after function was impaired by sleep deprivation. We then used it in a separate study to try to prevent the development of the deficit during a continuous course of treatment, so not just an individual rescue session, but actually a course of TMS treatment. we then used it in a series of studies to try to enhance resilience to an illness-related deficit. In this case, we're using as an illness model age-related cognitive decline. And ultimately, the next steps in this line of work in the field of aging is to try to remediate clinical symptoms of dementia and restore healthy function. And so you can see across this translational trajectory, we are going from healthy volunteer studies to clinical samples with an iterative approach in which we've focused on refining dosimetry and paying attention to all three aspects of dosage, the spatial, the temporal, and the context to optimize the intervention. My colleague Bruce Luber went through these studies in great detail. I'm just going to highlight the progression of the work to put it together in this bigger picture. So we started with enhancing normal function in this publication in healthy volunteers, where we gave the simultaneous TMS with working memory training and showed that we could enhance working memory performance in a fashion that was dependent on site of stimulation and dependent on the frequency. So that gets you our spatial and our temporal aspect of dosing. The next step that I'm highlighting here is the remediating experimentally induced deficit. In this case, we were targeting the circuit that was associated with resilience to sleep deprivation, and we gave our a combo intervention, which is the TMS plus working memory training, to remediate working memory performance deficit after 2 full days of sleep deprivation. And so that's basically still in the healthy volunteer space, but experimentally inducing a deficit and then rescuing it. And in that study, we were able to show the measure of target engagement, that the degree to which the active versus sham TMS improved cognitive performance, which was demonstrated by the change in reaction time on the task, correlated with the change in network expression, really allowing us to nail down that, that third aspect of target engagement to show that the behavioral improvement that we saw was correlated with a brain circuit function change in the predicted direction. The next step was a study to show that we could prevent the deficit from occurring in the first place. And in this experiment, we gave a course of treatment. It was 2 treatments per day for over a series of 2 days during the delivery of the sleep deprivation. And we found that when given in the site and frequency dependent fashion, TMS was able to prevent the development of the working memory deficit, and that improvement lasted a full 18 hours after the last TMS session was given. So this gives you a hint towards how you might design a trajectory of studies to ultimately apply this in a therapeutic setting with a course of treatment. And so now moving from the healthy volunteers to the clinical samples, the next stage is to enhance resilience in an illness-related deficit. In this case, actually, we're using age, healthy aging specifically. So these subjects do not yet have dementia, but they have age-related cognitive decline. This is a study that's currently funded by the National Institute of Aging. And what we found in this experiment was targeting the neural compensation network with our intervention, where we're coupling the TMS with the working memory training, was able to selectively enhance the working memory function in the elders. The sham is in the, the sham is in the black, the active TMS is in the yellow. That's vice versa. The sham is in the yellow, the active TMS is in the black. We were able to increase the percent correct in the elderly sample, and the same paradigm did not have the same effect in the young, so we consider this to be a compensation network. And then the next steps of this line of work is to try to remediate clinical symptoms as subjects progress towards dementia, mild cognitive impairment first and ultimately dementia in transdiagnostic samples, including other conditions where cognition is also affected. So I'm going to, since I know that my colleague Dr. Luber presented this task in detail, I'm going to skip over this, but only to emphasize on this slide that the timing is important. So the TMS was given specifically during the retention phase of this working memory task. And when we gave it during other phases, it either disrupted function or did not have a significant benefit. And so this highlights the importance of doing dose-finding studies early on so that you can figure out when to appropriately time your intervention, time lock with the task. Because if you just make a guess and do a large-scale clinical trial, you may finish your trial and not have an informative answer because you happen to pick the wrong stage. So while dose-finding studies are labor-intensive to do, we really do recommend doing that labor-intensive work early on in the course of study to inform where to go. So this is the design of our dose-finding study, where we examined two nodes within the network, the frontal cortex and the parietal cortex that was precuneus, and we gave either active TMS or sham on separate days to really figure out what is the optimal paradigm in space, time, and context. And within each day of stimulation, we had multiple blocks of stimulation where we ramped through different frequencies, 1, 5, or 20 hertz, during either retention phase or the probe phase of the task. So you can see with each day they were getting 6 blocks of stimulation, so 24 blocks of stimulation in all, so very time intensive. It took several weeks for each subject to complete the task. Within subject is important because of the individual differences in performance, and it was important to leave sufficient time between experiments to prevent carryover and to counterbalance the order to control for any carryover effect. What we found in that dose-finding study is that only during the 5 hertz conditions, in both the set size 1 and set size 6, so that's the relatively easy or low cognitive load and high cognitive load, active TMS in the blue bar significantly enhanced working memory performance as indicated by a reduction in reaction time relative to sham in the red. And this effect was specific to the parietal cortex site. It did not happen with the prefrontal cortex site. Contrary to our hypothesis that prefrontal cortex would be key for working memory, and certainly it's involved, but when we're looking at enhancing function above and beyond normal function, it turns out the area where we're not at ceiling, where we could actually drive the system in a way that it could improve, turned out to be the prefrontal cortex, turned out to be the precuneus, so parietal cortex. The next phase of the work then was to remediate cognitive performance after it was impaired by sleep deprivation. And in this study, we were stimulating this active site, which was part of the reserve network, and a nearby control site. And we found that when stimulating the active site, which was the superior occipital gyrus, we found a significant improvement as evidenced by reduction in reaction time, not seen with the nearby control site, which is the lateral occipital cortex. And also, we see now a context dependency. So the precuneus, which is this third black bar here, although that was effective in enhancing working memory performance in healthy volunteers who were not sleep deprived, it did not work at all following sleep deprivation. So really, this demonstrates the importance of context. This paradigm only worked when the subjects were sleep deprived. And then the last study here is the prevention trial, where 4 sessions were given over a 2-day period during the extended period of sleep deprivation. And so it was a randomized controlled trial, again, with that combo intervention of TMS given during time-locked working memory performance. And we found that the active group actually got a little bit better. The sham group got significantly worse, and this is a consequence, that's a normal effect of sleep deprivation. One of the measures we have about outcome is lapses in performance where the subject doesn't respond at all. It's as if they've had a little mini sleep. And lapses occurred almost exclusively in the sham group, not in the active group, and that was a significant difference. And to really nail down that the target was engaged and that the behavioral response was related to engaging our predicted therapeutic target, we applied E-field modeling to where we gave the TMS, which the TMS coil was placed right here underneath that blue circle, and you can see the E-field modeling shows that we were stimulating a region of parietal cortex which overlapped with the key node in the network that was actually changed by the stimulation, which was underneath the coil. So it demonstrates that we got a change, the in-network expression that was associated with behavior, and that change was accessible to, it was actually localized underneath where the TMS coil was placed. So as you're designing TMS studies for clinical application, there's some key decisions that you need to make about your methods. And I've highlighted some of these and given you some examples. The first is about the spatial aspect of dosing. So what is the optimal coil design, position, and orientation of the coil relative to the individual anatomy and relative to connectivity? And here are the tools that are important, or E-field modeling, structural MRI, DTI, diffusion tensor imaging for the tractography, and applying the human connectome to understand connectivity to distant sites. The second is the temporal aspects of dosing, the pulse shape, the terrain parameters, to optimize the effect. And I gave you an example where we tried 1, 5, and 20 hertz, and only the 5 hertz worked. The context, meaning using some manipulation such as a cognitive task to control brain state and to time lock changes in brain state to the TMS delivery. And then what sham or comparison condition you're going to use. I haven't touched so much on that, but I'm happy to answer questions on that. That's actually a huge issue for TMS study design, in part because we know that bias can affect outcome. We don't want subjects to be cued about what they're actually receiving. And also, TMS, when you get it, causes a clicking noise, it causes scalp sensation, and these ancillary effects of TMS are important, not just because they could cue subjects, but also because they can change brain activity as well. When you're hearing a clicking, your auditory cortex is going to light up, and that will obviously confound your imaging results, for example. And so, in conclusion, we have in the field of clinical application of TMS, illustration that spatial targeting of TMS to the dorsal prefrontal cortex is therapeutic in depression. And that concept of mapping the circuit and then using that as a map to apply TMS here can be productively applied to other conditions besides depression. Great areas of opportunity include identifying non-addictive interventions for the treatment of pain, identifying non-addictive interventions for the treatment of addictions, and just about anything that's related to a brain function that's disrupted in disease. If we can study its circuitry, we have the hope of intervening and remediating its circuitry using image-guided TMS or other tools. So while that has been successful in bringing TMS with depression to the field, to the stage of FDA approval, it has not yet been successful in any other condition, although research has been very promising in the treatment of other disorders, and some of my own work has focused on obsessive compulsive disorder, anxiety disorders, and I showed you examples from our work with memory. Temporal targeting, though, has been relatively less studied. So if we can understand where to give the TMS in the phase of different oscillations to enhance the expression of those oscillations which are functional, that have an influence on the operation of the circuit, this could improve the potency of TMS and the other interventions to treat these circuits to have optimal clinical outcomes. And this is an area that is relatively less studied, but has great potential for impact. And if you're thinking about where to invest in your research career, about where you might be able to make a difference and really educate the field about how to use these tools meaningfully, I would call your attention to this aspect of temporal timing and how to optimize the delivery of the TMS in time to modulate neural oscillations to have functional effects. This is relatively less studied. And ultimately, our understanding of how the endogenous neural oscillations interact with what we exogenously apply, which is the pulsed brain stimulation. That interaction, because the brain oscillation is going, tonically anyway, and how what we apply with TMS or other technologies modulates that and how they interact is going to be key to understanding not only how the technologies that we have approved today work, but also key to optimizing their application to improve brain health in neurological psychiatric disorders and addictions. So I'd like to recognize the members of the non-invasive neuromodulation unit, past and present. And I know that many of you have joined online. I've saved some time for questions, and I'm going to invite those now. If any of you online would like to unmute your phone and ask any questions verbally, or use the chat tool and send your questions to all, I'm going to pause here, and I'm happy to take your questions. And I do see a few of you online. I'm not hearing any questions. I'm going to offer a question that I'm frequently asked, which is, what else is TMS good for in terms of clinical application? I've talked largely today about depression, which is the only FDA-approved indication. I've also given some illustrations from experimental work with memory. We also have a significant evidence basis for potential therapeutic utility of TMS in other conditions. And this is supported by meta-analyses and reviews that really rate the level of evidence. And of the different applications where TMS has been applied, pain is actually one of them where there is significant evidence from well-controlled trials that TMS may be useful to somatosensory and primary somatosensory and motor cortex in the treatment of central pain syndromes. Another condition that has been studied is the negative symptoms of schizophrenia. This is a deficit area in terms of our medications, which are lacking in the treatment of the negative symptoms of schizophrenia. So when we think about where could TMS be fruitfully employed as a complement to our available medications, targeting things that the medications can't do is one example of that. There's also a sizable body of work in anxiety disorders, either comorbid anxiety and depression or primary anxiety disorders by themselves. Generally speaking, the low-frequency stimulation to the right dorsolateral prefrontal cortex has been showing improvements in both anxiety as well as depression. That's from some of our work and others using that paradigm. Obsessive-compulsive disorder, in the treatment of that, we've been focusing on the supplementary motor area, which is the part of the cortical basal ganglia thalamic limbic network that's disrupted in OCD. And there are other examples as well. I could say that there's probably more that we don't know about the clinical application of TMS than what we currently know, because it's a wide open field. And it's an exciting time to be in this field, and it's an exciting time to be working at NIH with the tools available to us to try to take this technology into the next decade, literally, to optimize their use. I'm going to pause again to see if there are any questions, either if you'd like to unmute your phone or use the chat tool. And if not, I'd like to thank you for joining us this afternoon. We're about at the end of the time. I'd like to make an open invitation. Those of us in the non-invasive neuromodulation unit at NIMH are happy to hear from you. If you are currently using these tools and have questions, or if you want to be using these tools and would like some help in terms of optimizing the dosing in space, time, and context, or in applying this in a psychiatric population, we are more than happy to help give advice, to collaborate, and to provide hands-on training. We offer training in the practical application of TMS and in study design. I also offer training in the safety aspects of TMS, so please feel free to join us for that. We also have a scientific interest group on non-invasive neuromodulation, otherwise known as the NIBS-SIG, and if you'd like to join the NIBS-SIG to be able to attend our speaker series that is held in Building 10 at the Intramural Research Program, please just shoot me an email at sarah.lisanby@nih.gov, and I'd be happy to hear from you. So again, I'd like to thank you for joining us, and good luck with your research, and I'm happy to answer any questions and follow up by email. Thank you so much.